Video to accompany the Dragon Lab that you are doing. This is to really describe the steps at step 5 to alleviate any confusion. At this point, we are going to simulate the crossing over that occurs during Meiosis I for a pair of chromosomes. For this, the two people in this lab that are partners will designate themselves partner A and partner B. This is separate from the mom and dad dragons. This is just really for us to kind of make the crossover process a little simpler. So you should have two coins. Here we have a penny and a quarter. Let's call the quarter our partner A coin and the penny partner B coin. And there are a set of rules here about what we will do with different coins. If it's chromosome 4 you're doing, note that it has a special rule because there are fewer genes on it. But for the other three, we're going to follow these rules here. So let's imagine that we flip our coins and we get heads on the first coin. Let's imagine we get tails on our second coin. If we look at that, then you'll notice here that partner A lands on partner A lands on heads, B lands on tails, and crossovers between the first two genes. So if we come to this, what we're going to do is we're going to break apart our chromosomes like so, and we are going to reattach them with the big A, little a swapped so that we now have recombinant chromosomes. You'll do this for all of your chromosome sets except for the X and Y chromosomes, which don't have more than one gene on them to cross over. And then once we've done that, we will be able to carry out our anaphase step, or actually our metaphase first and then anaphase. So the chromosomes will line up. Doesn't matter which way you line them up. We're going to flip a coin again. So here we have our two chromosomes. And you're going to do this for each one of the chromosome pairs. They'll all line up just like in metaphase of meiosis. And then you're going to flip one of your two coins. And so we'll flip that. And it landed on heads. So if it lands on heads, the rule says the coin lands on heads, then reorient the chromosomes in the pair. So what we will do is flip them around now. So now what was on the left is on the right, and what's on the right is on the left. You're going to do that for each one of your chromosome pairs. You're going to go through and flip a coin for each chromosome pair. If it lands on heads, you flip their orientation. If it lands on tails, you leave it alone. So that you have now thoroughly kind of remixed all of your chromosomes for each parent. So mom dragon will do this on her desk, dad dragon will do that on his desk, and we should end up with two sets of chromosomes at metaphase. Then you will separate them to their separate sides of the desk, and these will represent the two gamete pairs being produced. After that, you'll flip a coin again for which one gets to be the lucky winner that combines with a gamete from the other parent. And I believe it is heads of, let's see, well, it doesn't actually say, but let's assume heads is the winner. So if we flip this for this one, if it gets head, or whichever one, call this one heads and this one tails, you can have your own system. But if it's now tails, this one wins, and we would bring this chromosome 1 down to, or this chromosome pair, sorry, of the gamete to the other one from the other parent. So what you'll end up with is each partner in this lab will have a desk with two piles of chromosomes on it representing gametes. They'll then flip a coin to decide which one of those two will go and combine with the chromosome pair or chromosome set from the other parent who's doing the exact same thing. So you should have at that point then a pile of two separate chromosomes from the two separate parents, and each one of them will have a chromosome 1, chromosome 2, chromosome 3, chromosome 4, etc. This will give us our diploid set. Note that we are not dealing with doubled chromosomes in this lab, so there are no extra sister chromatids to deal with. There's no Meiosis II. We're simplifying the reproductive process here. Okay, hope this helps. This should get you through crossing over and independent orientation at metaphase and the separation at anaphase. So we've essentially mimicked the meiotic events here.